So it's now my pleasure to move on to our second keynote speaker of today, who is Dr. Jennifer Gommerman, who is the chair and also a professor in the Department of Immunology at the University of Toronto. Dr. Gommerman received her PhD in immunology at the University of Toronto, and then she went on to do a postdoctoral fellowship at Harvard Medical School, where she studied the complement pathway. She then moved out of academia and joined Biogen as a staff scientist. And during her tenure at Biogen, she became interested in B cells, multiple sclerosis, and the TNF superfamily of molecules. After her relatively short stint at Biogen, she returned to academia as an assistant professor at the University of Toronto and became a full professor in 2023, where she's now assumed the position of departmental chair. Her basic research continues to focus on members of the TNF superfamily of molecules that regulate both immunity and autoimmunity. Well, not both, immunity and autoimmunity. Her team of researchers have uncovered a novel gut-brain axis that regulates neuroinflammation. And with respect to translational work, Dr. Gommerman has been examining the role of B lymphocytes in multiple sclerosis patients, as well as in animal models of MS. Her contributions to understanding the mucosal antibody response to SARS-CoV-2 resulted in her co-leading biomanufacturing and pandemic preparedness efforts at various Ontario University. She's won multiple mentorship awards, is a distinguished fellow with the American Association of Immunology, and holds a Canada Research Chair in tissue-specific immunity. So without any further delay, I'd like to introduce a long-standing colleague, Dr. Jennifer Gommerman, and over to you, Jen, to please give your presentation to us all.
Thank you, Eleanor. Can everyone hear me?
Yes, we can.
Great. And you can see my slides and they are advancing, I hope.
Yes.
Great. Well, thank you for the invitation to speak at BSI. This is an incredible initiative that the Department of Immunology has been very proud to support in the past and to continue doing so going into the future. I think this is a major way to share science across the globe, and I'm really appreciative of Eleanor's efforts as well as all her colleagues who put this together. Thank you. So today I'm going to talk to you about IgA, my favorite antibody, and also I'm really going to focus on some work that's under review, not yet published. So this will be a great opportunity for some feedback. So I want to get everybody on the same page first and talk a little bit about what IgA does. So IgA is an antibody that's produced at mucosal surfaces, and it's produced as a dimer, and through this J chain that allows the IgA to dimerize, it binds to a polymeric Ig receptor expressed on the epithelium that allows the IgA to be shuttled outside of the body, where it can bind to microbes that are present in a particular luminal space. And I'm showing here the gut of the small intestine of the gut. And while we usually think of IgG as the workhorse of the immune system, it's plentiful in blood and it's important for neutralizing pathogens, especially viruses, through ADCC and complement-dependent mechanisms. This is really systemic immunity. In contrast, IgA is really about mucosal immunity, and despite it can't fix complement, and despite that, it's still very good at neutralizing viruses. And we tend to forget about IgA when it comes to protecting the host. And what I like to think of with respect to IgG versus IgA is that if your body is a dance club, then IgG is the enforcer who keeps the peace inside the dance club, so that if anybody gets out of control, they can kick them out. Whereas IgA is the bouncer that doesn't let potential threats into the dance club in the first place. And so that's one of the great advantages of IgA. And I think what we need to do moving forward with our vaccine strategies is figure out how to harness IgA responses at mucosal surfaces and make sure that those responses are durable. And that's a key. Now, the way IgA is generated is it is generated in what I like to think of as an induction site within the lymph nodes. And these induction sites can be, for example, the Peyer's patches of the gut or the mesenteric lymph nodes. And within these induction sites, class switch to IgA as well as IgG happens. But whereas IgG plasma cells typically home to the bone marrow, that's their effector site, if you will, IgA plasma cells make the long journey back to the mucosal tissue. And so in the case of the gut, an IgA plasma cell will leave the Peyer's patch, go into the efferent lymphatics, circulate through the thoracic duct, through the blood, and then all the way back into the gut. And within the gut, we actually know that plasma cells, IgA plasma cells, can live for a really long time. In humans, using carbon dating, it's been estimated that a lot of our IgA plasma cells in the gut can live for decades. So if we can figure out how to use this property, this becomes a really good vaccination strategy. And in fact, I'm showing you here a couple of cases where vaccines have taken advantage of IgA, long-lived IgA plasma cells. In the case of oral poliovirus vaccines, as well as rotavirus vaccines. And we and others have also shown that, although we haven't shown this causally, that IgA levels are predictive of whether a person may or may not get a breakthrough infection after a SARS-CoV-2 vaccine. So there's more and more cases that are emerging where IgA is important. Okay, so I'm really going to talk a lot about this détente between the induction site and the effector site, which in the case of the gut is the lamina propria. And I'm going to do this in the context of a virus called rotavirus. And I'm going to ask these 4 questions today. What are the kinetics of rotavirus responses? What is the longevity of this response? Does it protect the host against reinfection? And what variables make a difference to this response? And so this is the work of Dr. Kei Haniuda, a postdoc in my lab who joined me from Chiba, where he had been studying IgE plasma cells, and he wanted to look at IgA plasma cells in response to a virus at the mucosa. And we chose to study rotavirus. So this is an important gastrointestinal pathogen that is a leading cause of severe gastroenteritis in children. In lower middle income countries, this can be a devastating infection in children who are unvaccinated. They can lose a lot of fluid because of the virus, and so it's an ongoing concern. And the vaccines against rotavirus have variable efficacy depending on the populations they're tested in. But what we know for sure is that IgA is very important in protection with this vaccine. And we've received a strain of rotavirus from Harry Greenberg at Stanford that's been mouse adapted so that we can study all the different steps for how an IgA response to rotavirus is induced. And what Kay has done is he's really focused in on a couple of proteins that are expressed by the rotavirus and exploited these proteins to make tools so we could follow this response in vivo. And those tools up until now have been largely missing. So the first thing he did was he purified these 2 proteins, VP4 and VP6, and then he biotinylated them and used them to make tetramers so that we could look for rotavirus-specific B cells, and that's shown here. You can also use these proteins in ELISpot assays to detect plasma cells. But what I'm showing you here is flow cytometry data looking at the tetramers and looking at the kinetics of the IgA response within the Peyer's patch. And we see similar kinetics in the mesenteric lymph node. And what we find is that the IgA and the IgG response have very similar kinetics for the VP4 and VP6 antigens. But we did notice that there was a difference in kinetics when we used these proteins as ELISpot for ELISpot readout in terms of plasma cell generation. And specifically, there appears to be a more rapid IgA response in terms of plasma cell output that can happen quite early after exposure to the virus. And so when you look at the plasma cell output, IgA is actually faster than IgG. Okay, so what do these plasma cells look like? So to do this, we used these tetramers and we characterized the plasma cells by flow cytometry at very late, early and very late time points post-infection. And the first thing that Kay noticed was that as the infection proceeds, this is 180 180 days post-infection, you still see these plasma cells, but they're largely non-proliferative. And they have downregulated this surface molecule called TIGIT and upregulated another surface molecule called LY6A. And this is consistent with the phenotype of longevity that has been described by Hai Chi in 2022. So these look like long-lived plasma cells, and certainly they're long-lived based on how many days we can detect them post-infection. And this is that data summarized. So they seem to have a long-lived phenotype. What does the B cell receptor look like in these IgA plasma cells? So Kay sequenced several BCRs that are from VP4 and VP6 specific IgA plasma cells. And what he found was that over time, there was an increasing number of mutations in the CDR3 region, indicating an accumulation of somatic hypermutation activity. And what's interesting is that we know that this virus is cleared from the feces after as little as 10 days. And so here we are at 180 days post-infection, and this germinal set, what's likely a germinal center, is still percolating and resulting in B cells that have accumulated mutations. And if we look at those B cells, they do have clonal relationships to each other, suggesting that the process of affinity maturation is occurring within the Peyer's patches. So they've undergone somatic hypermutation and are likely undergoing affinity maturation. Is this IgA any good? And so what Kay did was, this is all a lot of work, but he was able to clone and express multiple IgAs that were coming from VP4 and VP6 specific B cells. And using some vectors that we got from Michelle Nussenzweig at the Rockefeller, he expressed these IgAs along with the J chain, the joining chain, to make recombinant dimeric IgAs. And when he tested these IgAs in vitro, he saw that they had a range of affinities, but some of them had very good affinity for their antigen, as you can see here. And he actually took some of these IgAs, these recombinant IgAs, and asked if they would actually neutralize the virus. So the way he tested this was he gave these antibodies prophylactically to B-cell deficient animals, and then infected these animals with rotavirus. And what he found was that there were several clones that completely prevented infection by the virus. There were some that had partial activity and some that had really no activity. So he saw a range, but many of them were able to protect against viral infection. This is our negative control dimeric IgA, which is a SARS-CoV-2 specific IgA. So we think this is sort of like sterilizing immunity. All right, so I've shown you that rotavirus-specific B cells make an IgA response, that affinity maturation in this IgA response occurs. And we know that this typically happens in a germinal center. And we know a lot in the textbooks for how IgG plasma cells are made. Briefly, what happens is that in a germinal center reaction, you will get class switch to IgG. And this is really dependent on two prior steps. The first step is that dendritic cells will pick up antigen and present it on MHC class II to T cells that are specific for the antigen. And these T cells can adopt a TFH phenotype. That is, they gain the ability to move into areas that are rich in B cells by expressing this chemokine receptor CXCR5 and this transcription factor BCL6. And once they're in B cell areas, these T cells talk directly to B cells, and the B cells themselves have picked up antigen or focusing that antigen to the T cells. And this allows for the germinal center B cells to undergo class switch as well as affinity maturation, because these B cells have an advantage within the germinal center. And then they leave as plasma cells. And as I said, if you're an IgG producing plasma cell, you'll likely go to the bone marrow. So we wanted to know if these steps were the same in an IgA response. And we started with TB interaction. And to do that, we removed MHC class II from B cells, and we asked if that would matter. And it does. And in fact, several aspects of TB interaction are required to make an IgA response to rotavirus. So here we eliminated CD40 signaling. Here we eliminated TFH cells. And here we deleted class II on B cells. And in all three of these cases, you shut down not only the IgG response, but you also shut down the IgA response. So far, this looks like a textbook T-dependent germinal center response. Okay, so then we did a control, and we went a step backwards, and we asked, well, what would happen if we change antigen presentation to T cells? So if we got rid of MHC class II on dendritic cells, and our prediction was that nothing would happen, and you would get no IgG response and no IgA response to rotavirus. But actually, we had a surprise. And so our prediction was correct for IgG. If you get rid of class II on DCs, you eliminate the IgG response. But when we looked at the IgA response, it was absolutely fine. If anything, it was a bit higher in mice that lack class II on dendritic cells. So this kind of blew our minds because that's not the way we've learned about B cell responses in germinal centers. You need T cells that have been primed by dendritic cells. And clearly that's not needed here. So we wondered whether this response was making IgAs that were any good. Maybe it's just all garbage IgA. But when we looked at the burden of mutations in the B cell receptors of these IgA positive B cells that had been primed in the absence of class II on DCs, it was just the same as the wild type mice. And these IgAs can protect against infection, as shown in this particular experiment where we used that IgA to, we mixed it with the virus prior to infecting, and that prevented infection. So these IgAs are hypermutated and they seem to block the virus. So this is saying that expression of MHC class II on CD11C positive dendritic cells is not required for a rotavirus-specific IgA response. So then we went a step further. We knew that you needed class II on B cells, but we wondered if maybe that's all you need. Maybe in the case of a rotavirus IgA response, you sort of kick out the middleman. You don't need them. You don't need dendritic cells. So to ask this question cleanly, we turned to mice that we got from Greg Wu at Washington University that express MHC class II only in B cells. And if you use these mice, you have to give them some T cells because thymic selection is defective in these animals. But once you give them T cells, they're just fine. And so we used these and asked whether these mice could make an IgA response to rotavirus or an IgG response to rotavirus. And so there's three groups of mice here. There's the positive control where MHC class II is expressed normally. There's the negative control where there's no MHC class II, but they got some T cells. And then there's our experimental group where we've allowed expression of MHC class II only on B cells, but again, we've added supplemental T cells. For the IgG response, we get exactly what we would expect. That is that in the absence of class II or in the presence of class II only on B cells, you don't get an IgG response. You really need class II on dendritic cells. But for the IgA response, while you certainly need class II, having class II on B cells is actually sufficient for the VP4 and the VP6 specific IgA response. So this is unusual. This means that you can really prime this response just through B cells. So we started to think, well, how are these T cells getting primed and what do they look like? And we haven't fully answered the first question, but I will give you some data to examine the second question. So what do these T cells look like that help a rotavirus-specific IgA response? And we postulated that there might be a conventional TFH that helps the IgG response, which unrolls in the same Peyer's patch germinal center as the IgA response. But the IgA response might need a different type of follicular helper cell, and that this would be to get the IgA response going, follicular helper population. And so that's just summarized here. So, but the problem is we didn't have any tools to really TFH cells that are specific for the class II restricted peptides that are present in VP4 and VP6 proteins. This was a long process where he screened many, many peptides, but he was able to find some peptides that had really good binding motifs for class II. And in collaboration with colleagues from University of Minnesota, tetramers were made so that we could detect these T cells. And we found a couple of tetramers that worked really well. So now we have a great tool for fishing out rotavirus-specific helper T cells. And we use those tetramers to map the T cell response at the single cell level. And so this is single cell RNA sequencing of Peyer's patch and mesenteric lymph node T cells. where we've used tetramers. And this is where the tetramer positive cells map back onto the T cell populations. They're primarily in the Th1 and TFH populations, which is what you would expect. We know that this response is TFH dependent, and it's a virus, so we know there's going to be a lot of interferon gamma, in other words, a lot of Th1 cells. But one thing that we saw when we reclustered this data to only look at the tetramer positive cells was that there was actually two populations of TFH, and these were transcriptionally distinct. And one of these populations, population 6, was a TFH that also expressed TBET. That's a transcription factor that's associated with interferon gamma production. And they also expressed interferon gamma. So this is weird because usually TFH and Th1 they're sort of in détente. And here we have a cell that's sort of halfway in between. And actually, this is a fairly stable population because we were able to see these TFH1 that we gate here using this Th1 dependent chemokine CXCR3 and a reporter for TBET that we got from Sasha Rudinsky that reports TBET expression with TD tomato. And when we gate on antigen-specific TFH cells, we see this population and it persists. So this isn't just some fly-by-night weird thing. This is something that actually sticks around for the whole viral response. So we wanted to learn more about these cells. We characterized them a bit more. It turned out that there were some, especially at the early stages of the immune response, that were negative for CD90. And these CD90 negative TFH are thought to be the ones that are actually in the germinal centers. We thought that was interesting. And if we delete MHC on class 2 on B cells, which we know is important for the rotavirus-specific IgA response, those TFH are gone. So these appear to require cell-cell communication with B cells. Moreover, if we look at the mice that have MHC class 2 only on B cells, what we find is that these TFH1 still are produced. So that's consistent with what we found when we looked at the IgA response in mice that had MHC class 2 only on B cells. So this all fits together. All right, so antigen presentation by B cells seems to be necessary and sufficient to generate these CD90 negative TFH1 that are specific for rotavirus VP4 and VP6 peptides. But are they actually needed? So to test this, Kay got rid of these TFH1 cells. And the way he did this was he used this TBET-CRE that he knew was expressed in TFH1, and he deleted this transcription factor BCL6, which is necessary for TFH identity. So in this cross, what he's done is he's gotten rid of TFH1. And what he found was that when you get rid of TFH1 cells and you look at the two different responses, the IgG response and the IgA response, the IgG response doesn't care. It doesn't need TFH1 to develop. But the IgA response is greatly inhibited without TFH1. So this means there's something special that TFH1 are doing to allow for an IgA response. Now, I'll point out that this is at the effector site. So we went back and we looked at what could be going on within the Peyer's patch itself. And our hypothesis was that within the Peyer's patch, where this immune response is developing to IgA, that TFH1 are making interferon gamma, and that this interferon gamma is required to somehow license these IgA switched B cells to go to the right place. That was our hypothesis. So we tested this by eliminating interferon gamma in the TFH1, and we also eliminated interferon gamma receptor signaling in the B cells, and I'm going to show you both of those results. So when you eliminate interferon gamma in the TFH1s, and I won't go into how we did this, but I'm happy to discuss it afterwards, but this was a sort of complicated mixed bone marrow chimer we had to do. What we found was that, like what we saw when we got rid of TFH1 altogether, if you lack interferon gamma production by TFH1, you only lose the IgA response and not the IgG response. So that's a phenocopy with what happens when you lose TFH1 responses. So interferon gamma production by these T cells is very important. So what could interferon gamma be doing to the plasma cells? And one thing we thought about was that either this is somehow needed for the expansion of these IgA plasma cells, maybe the generation of these, the expansion of the germinal center B cells, or perhaps the generation of the plasma cells, or perhaps the accumulation of these plasma cells at the effector site, which is the lamina propria. So we aim to test these possibilities. And if I can move my slides forward, we did another one of these chimeras, but here it's a little bit different. What we asked was if we take B cells, half of which are gamma receptor sufficient and half of which are interferon gamma receptor deficient, who wins when we put them head to head? And so we did this chimera experiment. And what we found was that in the germinal center in the Peyer's patch, it didn't matter whether a B cell expressed interferon gamma receptor or not. It still is generated. You can make an IgG positive germinal center B cell, and you can make an IgA positive germinal center B cell to either antigen just fine. If you look at the plasma cells that are initially generated in the Peyer's patch, likewise, there's no defect. The chimerism is practically 50-50. When you look at the effector site, so for IgG plasma cells, this is in the bone marrow, you see no impact of interferon gamma receptor on the accumulation of IgG positive plasma cells in the bone marrow. But when you look at the accumulation of IgA positive plasma cells in the lamina propria, now we see a very dramatic defect. And so this suggests that where interferon gamma receptor is acting is in that step where plasma cells move to their effector site, because that's where we're seeing the defect. So this is okay. The germinal center is okay, but this migration step is not okay. We looked at some of the canonical receptors that we know are important for this migration event, alpha-4 beta-7, CCR9, and they were expressed normally on IgA plasma cells. But if you remember, we were also using this marker called CXCR3 to define, in this case, TFH1 cells that had been primed in the presence of interferon gamma. And so we wondered whether CXCR3 might also be expressed on plasma cells, IgA plasma cells, as a consequence of their exposure to interferon gamma from TFH1 cells, because CXCR3 is a target of interferon gamma receptor signaling. So we went back to these mixed competitive bone marrow chimeras, and we asked whether having a gamma receptor was needed to upregulate CXCR3. And what we found was that, first of all, plasma cells that are rotavirus specific do express CXCR3, but this is impaired if you lack the gamma receptor on those plasma cells. And moreover, the abundance of these plasma cells within the gut is reduced within the Peyer's patches, but also within the lamina propria itself. And this is particularly evident. It's evident at day 14, but it gets worse and worse as time goes on. So here's day 70. But in contrast, the IgG plasma cells in the bone marrow are just fine. So what I've shown you is in this study is that what happens is that there's some kind of weird priming event where you don't need dendritic cells, and T cells acquire this TFH1 phenotype. They go on to help B cells and license IgA positive B cells via their production of interferon gamma to go to the lamina propria, where they remain for quite a long time because we can detect them as many as 180 days post-infection. So is this just a rotavirus thing? And this is my last data slide. The answer is no. So we asked whether some of these findings might be recapitulated in the context of an airway virus. And here we turned to influenza, and we used the mouse-adapted PR8 influenza. And the first question we asked was whether influenza infection through the nose, not orally like rotavirus, but now breathed in by these animals, whether that could lead to a long-lived IgA plasma cell response. And that's exactly what we found. So if we look for these influenza-specific IgA plasma cells, we can see them in the nasal mucosa and in the lung and in the bone marrow, and we can see these as late as 180 days post-infection. And we see IgGs as well. And this was seen by Akiko Iwasaki's group as well, so this seems really a characteristic of flu infection. We looked for TFH1 in the context of influenza priming in the mediastinal lymph node, which is the lymph node that drains the lung. So this is kind of like, the equivalent of the Peyer's patch for the gut. And what we found was that we could detect antigen-specific T cells that were TFH, as you would expect, And some of these TFH, actually quite a few of them, were expressing TBET. So these were TFH1s, and the rest were not expressing TBET. So there's a big population of TFH1s as well as conventional TH1s that emerge from influenza infection. And if you remove these TFH1, the same way we did in the rotavirus infection, we did see a selective defect in the IgA response in the nasal mucosa and in the lung, but not in the bone marrow. So there could be a role for licensing these B cells via TFH1 to go to their these sites, to the nasal mucosa and the lung. But the phenotype wasn't as dramatic as we saw with rotavirus. So it could be that this is a partial effect and other T cells can compensate. The other thing that diverged from our rotavirus findings was that if you delete MHC class II on all cells except B cells and give exogenous T cells, like we did before for rotavirus, in this case, the IgA response is not supported. So that seems to be different. In the case of rotavirus, MHC class II expression on B cells is sufficient to elicit an IgA plasma cell response. In the case of flu intranasal, it's not sufficient. You do need other cells to express class II. Okay, and so I'm going to finish there with this story of how we make IgA responses to mucosal infections. And I'm happy to take some questions. This is the lab and our funding. And we've had many collaborators help us on this project. In particular, I want to acknowledge Kay, who really led this work, and others who have helped, especially Mark Jenkins and Dilipan at University of Minnesota, who made the tetramers, Greg Wu, who made the mice, and other reagents. Thank you.